Hello, students, and good evening. Myself, Harun Rashid. So, today we are going to discuss our second lecture. In our first lecture, we discussed about genetics and the background of genetics, and today we are going to discuss a very important law of genetics, the law on which genetics is based. Yes, we are going to discuss Mendel's law of inheritance. So these laws have two parts. Today we are going to discuss first part. Then in our next lecture we are going to discuss this second part. So I am going to share my screen here with second law of Mendel's and reading, and you now speaker Harun Rashid. I am Harun Rashid. So contents. What we are going to discuss: Mendel's law of inheritance, and we are going to discuss key terms, and also we are going to discuss law of segregations. So this is our first part of Mendel's law. In our second law we are complete discuss remaining parts of this law. So Mendel's law of inheritance, basically who's the Mendel's law of genetics laid the foundation for understanding the principle of inheritance. Yes, Mendel's who's laid the foundations, how's these inheritance, how's the characteristics are transferred from parents to offspring. George Mendel's an Australian monk. The who's monk basically is the person who practices religious practices, okay, practice religious. So conducted experiment with pea plants. Mendel's conducted experiment on pea plants in mid 19th century, and his work became the basis for modern genetics. So in Mendel's law we are going to discuss some terminology. So first is dominance and recessive. You guys, dominance and recessive. What is dominance? Dominance basically is dominant allele mask the expression of recessive allele in heterozygous individual. So I am going to tell you what is dominant allele. Guys, you can see here, this is YY. YY is basically a genetic makeup of pea plant. So this is Y and this is Y is allele. This Y is show the yellow color, and this y show the green color. So this is female and this is male. For example, if they cross each other, from this one is one y come here, and from this one is one small y is come here. So they are come here, and here you can see here is yellow color. So this y, capital Y, mask the small y characteristics. So this is called dominant allele. So in heterozygous, this is heterozygous, capital Y and small y. Heterozygous is basically two different allele. And same like this is homozygous, homozygous dominant allele. This is homozygous recessive allele. So this is the dominant, you can understand the dominant, and homozygous, dominant and heterozygous individual express the dominant trait. So so this is the heterozygous dominant, and this is the homozygous dominant, and this is the recessive. So this is the recessive allele which is going to mask their characteristics due to dominant allele. Why? So I hope you can understand the concept of dominant and recessive. So we are going to our next slides. This is the some key terms which is involved in uh, Mendel's law. So first of all is genotype. What is genotype? Genotype refers to the genetic makeup of an organism. So you can see here, this is the genotype is genetic makeup. YY is yellow color of pea plant is a genetic makeup of a pea plant. So this is the phenotype. Phenotype is observable physical or biochemical expression of the genotype. You can see here, this is the genotype of organisms of pea plant, and you can see yellow color. Yellow color is basically a physical characteristics which are appearance able. So we can see this characteristic. This is called phenotype. And now monohybrid crossing plant that differed in only one trait, only flower color. When we are going to cross monohybrid, so we are going to discuss only one trait. We are going to discuss flower color. Okay, result in 3 ratio 1 3 1 3 ratio 1 of dominant to recessive phenotype in F2 generation. So we are going to discuss our next slides what is 3 ratio 1. And dihybrid cross. Dihybrid cross basically cross plant that different in two traits, flower colors and seed shape. So these are dihybrid. In monohybrid, we are just discuss only one trait. Monohybrid cross में sorry in monohybrid this is my language monohybrid cross में so sorry in monohybrid cross we are discuss only one trait flower color and in dihybrid cross, dies mean guys two. Students, you can understand dies mean two. So dihybrid cross, we are going to discuss two traits of plant, flower color and seed shape, and resulted in 9 ratio 3 ratio 3 ratio 1 in the F2 generation. Yes, we are going to discuss this ratio in our next part of our this Mendel law part. So in our today part, we are going to discuss 3 ratio 1 part. And here is dominance. A dominant allele is one that, when present in an individual genotype, genetic makeup, will express its characteristic traits. We can discuss here. This is the dominant allele. Y. Capital Y is dominant allele in heterozygous because he express their characteristics and mask the small one. So this is dominant allele. And now recessiveness in heterozygous individual, the recessive allele remain hidden. Okay, remain hidden or unexpressed in the phenotype due to presence of dominant allele. So you can understand here. Here is why is remain expressed because here is yellow color in F1 generation. If we cross two traits, yellow color plants and green color plants, so in F1 generation we get yellow color because this is dominant allele, and then we recrossed F1 generation, then we get variations. So what we get variations we are going to discuss in our next slide. So hope you guys can understand this simple scheme because these are very necessary to understand the Mendel's law. So here is law of segregation. First law of segregation, Mendel's. What is first law of segregation? Each individual has two factors allele, yy, small y, and capital Y, capital Y. This is two alleles. You can see here, small y is two allele. For each trait, for yellow color, they have two allele. For green color, they have two allele. One inherited from each parent. So when they are crossed to each other, one from this one and one from this one. So it's mean when two traits are crossed each other, each trait have one alleles go for crossing. Okay, from there is one and from there is one. Okay, these alleles segregate during gamete formation, and each gamete carries only one allele for each trait. Okay, so these are come and this is F1 generation. When these F1 generations are recrossed, okay, when this F1 generation are recrossed, then they segregate and give the variations and give and only one allele for each trait. Each trait have one allele. So here we discuss about what is this. This is Punnett scale. You can see here this is Punnett scale. So guys, you can students, you can see F1 generation. This is F1 generation. Okay, when we cross this F1 generation between each other, capital Y small y cross capital Y small y. So here you can see capital Y small y crossed capital Y small y. So when this capital Y cross this capital Y, we get two dominant allele. This is yellow color. Okay. When this capital Y cross this capital Y, sorry, when this small y cross capital Y, we get heterozygous. This is also called a yellow color because this is dominant gene allele mask the recessive allele. So when this capital Y cross small capital Y, this is also get heterozygous. Okay, this is also these are yellow color. And when this small y cross small y, we get green color. So guys, we how much we get yellow color? 1, 2 and 3. Okay, 3, and we get only one green color, ratio 1. So here we discuss 3 ratio 1. Okay, so when these are recrossed, so we get 3 ratio 1. This is called law of segregations when they crossed F1 generation when crossed each other. So uh, and each gamete carries only one allele for each trait. So what is Punnett square? Punnett square a tool. Basically, this is the Punnett square. Okay, you can see this table is Punnett square, a tool used to predict the outcomes of genetic process. So here we predict outcome. So what we get? Yellow color, green color, yellow color, green color. This is tell us the outcome of genetic process. Show the possible combination of alleles in of offspring. Yes, we discuss here possible combination. This y cross this y, combination. This small y cross this y, possible combination. This y cross small y, possible combination. So in Punnett scale basically tell us the outcome of genetic process and also tell us the possible combination of allele or offspring. So here is this is all about our first part of Mendel's law. Hope you are understand what is this, and in our second part we are going to discuss 9 ratio 3 ratio 3 ratio 1. So hope guys you are understanding this Mendel law of inheritance, and hope you are enjoying my lectures. If you are enjoying, if you are getting little bit information about my lecture, then please click the like button, and also please share your comments with me, your your thoughts, your feedback, and my fault, everything you can share in comments box, and I will improve this. And if you want to give any lecture on genetics and breeding, here is my email. You can contact with me, and then I'll record your lectures about genetics and breeding, and then we will post it on my channel. So next we will discuss about second part of Mendel's. So take care and wait for my second lectures.